Hello, I'm Weston. Today I'm going to be talking about DNA replication. So first, DNA, you can see here it has a double helix structure. And to divide it up into two strands, the helicase enzyme has to move through here and cut the hydrogen bonds. While here there's a topomerase, which prevents the dividing from going past and also preventing the uncoiling from going too far. So first we're going to talk about the leading strand of the DNA. It goes from the 3 to 5 direction towards the fork, while there's the lagging branch, which goes from the 5 to 3 direction towards the fork. So with the leading branch, it's very straightforward. The DNA polymerase simply just moves towards the fork and puts in the nucleotides. With the lagging branch, it's more difficult. So first, the primase has to be set in place so it can put down the RNA primer. This RNA primer allows for the DNA primase to go towards this direction because it cannot move from the 5 to 3 direction towards the fork. So it just does this and puts in these nucleotides. And then the primase moves on towards the next fragment called an Okazaki fragment. This fragment made right here. And it sets up the next RNA primer, while the ligase moves in and patches together the two fragments with the already replicated DNA. And then this process just repeats again and fills in these nucleotides.
Transcription is the process by which DNA is rewritten and encoded into RNA. It occurs in the cytoplasm of bacteria in the nucleus of eukaryotes. Much like DNA, RNA consists of a phosphate backbone and nucleotides. However, the phosphate backbone consists of alternating phosphate groups and ribose sugar, compared to deoxyribose sugar in DNA. RNA polymerase is the enzyme responsible for transcription. It binds to the promoter, a section of DNA at the beginning. This binding is aided by transcription factors, which are proteins in eukaryotes that help the RNA polymerase bind to the promoter. The RNA polymerase then acts much like helicase and is able to unwind the DNA sequence. It then moves downstream from the 3' to 5' end of DNA and reads the DNA sequence, adding nucleotides to the 3' end of the RNA sequence it is constructing. The base pairs are the same as in DNA, however, the uracil replaces thymine when pairing with adenine. Once we are done pairing up our base pairs, we now have our RNA sequence. In eukaryotes, there's an additional stage of mRNA processing, with the addition of a 5' cap and poly A tail, as well as splicing out introns, leaving only the parts which will be translated into amino acids. The mRNA then moves on to translation, where it can be read into amino acids.
Hi, I'm Kevin, and I'm going to be doing the translation portion of our project. So, here is the strand of template DNA, and then up here is the mRNA strand for the template DNA, which is just the same thing except the complement of it, and in RNA and anti-parallel, so the 5' and 3' ends are reversed. And from the codons, we can determine that the amino acid sequence will be, in order, methionine, phenylalanine, proline, alanine, lysine, and then a stop codon, which doesn't go to amino acid, but just makes the ribosome release a peptide. Initiation is the first step of translation. What happens is the small ribosomal subunit will bind to the mRNA, and then a special initiator tRNA will bind to the start codon, and the start codon, because it's also the codon for methionine, the tRNA will have a methionine bound to it. And the way the tRNA recognizes the codons and binds to them on the mRNA is through an anticodon. So this end of the tRNA has an anticodon, and it's just a complementary sequence to whatever the codon on the mRNA is. So for example, for the UUU here, the complementary sequence and the anticodon would be AAA. So once the tRNA is bound, the large ribosomal subunit will bind to that and complete the initiation complex. So the next step of translation is elongation, in which the rest of the amino acid sequence is completed. So the tRNAs will come in, and then they will recognize the codons on the mRNA, and they do this also through codon and anticodon recognition. And then there are amino acids on both of these tRNAs, and what happens is a peptide bond forms between the amino acid attached to this tRNA and the amino acid attached to this tRNA, and then the peptides, the amino acids are transferred over to this tRNA, and then this tRNA is removed, and then the cycle continues with each new codon in the mRNA, with each new tRNA coming in, taking the amino acids bound to the previous one, and then the previous tRNA being removed and replaced by the new one, until all of the amino acids have been bound to each other through peptide bonds. And then once that happens, is termination happens. So once a stop codon is reached in the mRNA, a release factor will bind to the mRNA instead of a tRNA, and that will trigger the ribosome to release the free polypeptide, and the whole structure of the ribosome and the translation complex will fall apart. And a lot of the work here is powered by the hydrolysis of GTP. Yeah, and in prokaryotes, translation can happen during transcription, so it happens like right next to where transcription happens because there's no nucleus, and in eukaryotic cells, translation has to happen in the cytosol away from where transcription happens because the nucleus separates it. Or it can happen in the ER.
Hello everybody, today I'll be talking about mutations into the DNA sequence. The first kind of mutation I'll be talking about is point mutation, also known as substitution. And into this process, it's a gene mutation that causes either a single or multiple nucleotides to change into the DNA sequence. And so visually you can see right here, if I remove this thymine and add a cytosine, you can see the mutated DNA sequence. Within point mutation, there's three kinds of different mutations. The first one is silent, which has no effect on the protein sequence at all. The second one is missense, which results in an amino acid substitution. And the third one is nonsense, which substitutes the stop codon for an amino acid. The second kind of mutation that I will be discussing is insertion. And so inside insertion, this is when a nucleotide is inserted into the DNA sequence. And so right here, you can see I have a visual representation of me inserting. And so right here, what happens is there are too many. You can obviously see there's 5 on the bottom and there's 4 on top. So what happens is there's a frame shift. So everything moves over 1. And so within this frame shift, it can change the whole entire codon sequence. And so the last kind of mutation I want to discuss is deletion. And so inside deletion, if I move this back to how originally it was, in deletion, it does the exact opposite. And so what happens is that one of the bases is straight up deleted. So if I remove this adenine right here, then what happens is a frame shift occurs for this deletion. So this adenine would move over here, but then you see this guanine would also move over here. And so, yeah, those are the mutations.